All right, welcome everybody. Thank you very much for joining us today. We've got a little message from Dr. Hank Paulson that I'd like to play for you all.
Hi, I'm Hank Paulson, Director of the Michigan Alzheimer's Disease Research Center, and I wanted to welcome all of you to this orientation meeting for the research education component. I'm sorry I can't be there. I'm away at a Gordon Research Conference. But I did want to say how important this part of the ADRC is. The REC components are a key element of any ADRC. They're how we can facilitate the training and the future career development of people interested in dementia-related research. And you are those people. You have a great group of leaders in Peter Lichtenberg, Irving Vega, and Roger Albans. And you have the core leaders across the entire center in the biomarker, neuropathology core, imaging core, data core, clinical core, who you can leverage as you continue in your work. I'm always available. Please reach out to me too, even though I'm not there today. I'm always happy to hear about what you're doing and give you advice if I can about your own career. Anyway, welcome to the REC. We're glad that you're part of our program, and we're very excited about the curriculum that Roger, Peter, and Irving have put together for you in this year. Thank you.
Great, and I should say truth in advertising, our real leader is Roger Albans. So Roger, I'm going to turn it over to you.
Okay, well, very good. Thank you very much, Peter. Yeah, thanks to everybody for joining. So just a couple of words about this. So the purpose of the session today is just for us as in the ADRC to get an idea of a little more detail about what you guys are doing, so that hopefully we can help you with your projects and career progression, and also just to give you some familiarity with the scope of the resources that are available through the ADRC, which we hope you'll be able to utilize for in your research. So with that, I think why don't we go to our first speaker? I think it's Andrew Brewer.
First, we're going to do introductions of faculty and staff that are here.
All right. So actually, I'm just going to correct Peter. Peter said I'm the leader of the REC. No, actually, Stephanie is the leader of the REC, actually. So go ahead.
All right. I'm just going to go across my screen. I guess first I'll introduce myself. My name is Stephanie Nava. I am the REC manager. You'll be hearing from me when you need different things, different things that are due, your progress reports. If you have any questions about REC, you can reach out to me and I'm happy to find answers for you. I'll also be, contacting you throughout the year with different funding opportunities and things that you might want to take advantage of. So you'll be hearing a lot from me. I think I'll just go across my screen. Peter?
Yeah, welcome everybody. Peter Lichtenberg. In addition to co-leading the REC, I am at the Institute of Gerontology at Wayne State University. I'm a clinical geropsychologist.
All right. Renee.
Hi, everyone. I am Renee Gadwa, and I manage the center's outreach, recruitment, and engagement core. So our core is responsible for all of the recruitment of participants into our research registry. We call it the Mindset Registry, as well as all of our center's events and communications and such. So I'm sure I'll be in touch with you over your time as a REC mentee, and nice to meet you all.
All right, Roger.
Hi, so Roger Albans, I'm the nominal director of the REC. I'm a movement disorder neurologist. My primary research area is actually Parkinson's disease. And no, I'm very pleased to be able to learn some more about your research today.
Anneliese.
Hi, everyone. I'm Anneliese Filipiak. I am part of the clinical core through the Leaders Initiative, and that was something that occurred after my own REC menteeship, I think 2019-ish. So happy to talk about the benefits of this program. I am a neuropsychologist in the Department of Psychiatry. With Clinical Core, I head up our feedback program, so providing return of research results on an individual basis to our participants in the longitudinal cohort. And my own research is looking at health disparities, particularly in how we diagnose and treat Alzheimer's disease and related dementias recently has been more focused on culturally informed approaches to biomarker disclosure. Really happy to be here and excited to meet all of you.
Doug.
Yeah, hi everyone. Doug Knoll. I'm in the Department of Radiology and Biomedical Engineering here at University of Michigan. I co-direct with Ben Hanstead. I see he recently came online. The Neuroimaging Core. We collect MR images on a couple hundred members of the longitudinal cohort every year. We also, either through direct funding of the ADRC or other affiliated R1s, there's quite a few PET studies also done, and we can help get access to that data if it's relevant for your research. And I'm actually looking forward to hearing what you're all doing and learning more about you.
Sharia.
I'm also part of the Leaders Initiative and work with the Outreach and Recruitment Core. I'm Sharia Robinson Lane, faculty in the School of Nursing, and my work is mostly focused on family caregivers of persons with different types of dementia.
Cindy.
Hi, I'm Cindy. I'm really glad Doug Knoll went first because he explained what the neuroimaging core is. So I manage the neuroimaging core. So me. And then everything that Doug said, just insert there.
Kelly.
Hi, folks. My name is Kelly Bukulski. I'm faculty in epidemiology at the University of Michigan in the School of Public Health, and our research group studies environmental chemicals and genetic factors that contribute to risk of Alzheimer's disease. I'm the leader of the data core here at the Michigan Alzheimer's Disease Research Center. I work closely with our manager, John Reeder, where we attempt to integrate all of the exciting data that's generated across all the cores of the center, harmonize it and put it in a format that we are able to share with you all. So we'll work closely with particularly those of you in the REC who are interested in working with some of the existing or ongoing data that the center generates.
Ari, now that you started drinking your water.
Good afternoon, everybody. My name is Ari Pomik. I'm one of the two administrators here at the Michigan Alzheimer's Disease Center. I co-administer the center with Nancy Laracy. She's on a well-deserved vacation right now, climbing the sand dune somewhere in Empire. My role primarily is as that of a research administrator. So if you need data, access to data, you need IRB stuff, MTAs, you need your rec funds, you would reach out to Stephanie and Nancy and me, and I would be that conduit to help you along there. And if I don't know the answer, I'll ask the right people. So, welcome.
John.
Hi everyone, I'm John Reeder. I'm the data systems manager here at the MADC, working with Kelly Bukulski. And so anytime you're interested in data from the center or analytical support, we'll make sure to get you the right person on the team.
Nick.
Hi, everybody. I'm Nick Cannon. I'm a professor of translational neuroscience at Michigan State University. For the center, I lead the biomarker core along with my co-lead Dave Morgan over here in Grand Rapids. So the biomarker core is actually based out of Grand Rapids. And we're currently measuring sort of these core AD biomarkers in plasma. So some phospho, tau, amyloid, GFAP, among a few others for the center, and working on developing novel assays. Traditionally, my research focus has been on Alzheimer's disease and other tauopathies with a focus on tau protein biology and pathobiology. So nice to see all of you.
And Ben.
Hey, Ben Hamstead. Let's see, wear a couple of different hats with the center. As you heard, the imaging core co-lead with Doug, and then Judy Heidebrink and I lead the clinical core. So pretty much all the data that any of y'all may access has come through clinical core at one point or another. And then my own work focuses on non-farm interventions. We're doing a lot with brain stimulation and integrating structural and functional neuroimaging and more PET scans, thanks to connections with Roger and others. So welcome, everybody.
Right. Thank you very much for taking your time to introduce some of our key staff here. Let's go ahead and jump right into our presentations. Our first presentation today is by Dr. Andrew Brower.
Great. Thanks so much. Thanks, everyone. I will feel free to interrupt me if you have questions. Not sure if this will be 20 minutes otherwise, but hello, I'm Andrew Brower. I'm faculty in the Department of Epidemiology at the University of Michigan. I've been research faculty for a number of years, and I'll be switching over to assistant professor in the fall. I'm a computational epidemiologist, and I work broadly in infectious disease, cancer prevention, and tobacco control, and now hopefully also dementia research. So my project is called Determinants of Transitions in Alzheimer's Disease, a multi-state transition modeling approach. But before I get into that, I want to tell you a little bit about myself. As I said, I'm a computational epidemiologist. My PhD is in applied mathematics from here at the University of Michigan. I'm originally from the North Country in New York, and my research interests are really largely around mathematical and statistical modeling in public health. Like I said, I've worked a lot in infectious disease, cancer, tobacco control, but kind of really anywhere that there's data, I'm kind of interested in understanding mechanistic reasons underneath the dynamics that we see. So I work a lot in longitudinal methods, including transmission modeling. I also work a lot with identifiability analysis and uncertainty quantification. So really understanding, based on data we have, what can we say about it? What can we say with confidence? Multi-state transition modeling, which is what this presentation and my proposed research is about, and things like age period cohort analysis, looking at different ways time can affect dynamics in our data. Also, just a little bit about me outside of work. I have a small homestead and small orchard, so I do a lot of gardening, beer brewing, jam, beans, garlic. So just to help give a little more context behind the face. So to come into my proposed project, as you are all familiar, dementia is a progressive disease, but that's really kind of the basis of how I think about the work that I'm proposing. So thinking about dementia as staging from kind of preclinical disease to mild cognitive impairment to clinical dementia. And if we could better understand progression between these stages, we could improve clinical care. We can develop personalized medicine approaches that could include things like individually projecting possible future trajectories and this associated uncertainty. So kind of given one's current state of imaging and biomarkers and impairment, what is that? What are the kind of prognosis kind of looking forward, and what's the variability that we might see? That's at a very kind of individual perspective, but on the kind of population scale as well, we can think about making population scale predictions of the future healthcare burden to kind of understand, as our population is aging, what can we expect to see over the next coming decades? What does that look like from what would be appropriate from kind of healthcare policy and so on and so forth. That's kind of the rationale for why do what I'm proposing to do. And so I'm really thinking about how progression is associated with things like individual characteristics, biomarkers, cognitive testing, neuroimaging, and kind of many other metrics that I'm hoping to learn about from everyone here. To really understand progression, we need to analyze longitudinal data. And so the longitudinal cohort associated with the Michigan Alzheimer's Disease Research Center is kind of the reason that I was really interested in pursuing this. And so here's just kind of a schematic graphic of what longitudinal data might look like as people have different kind of patterns of different trajectories, different patterns of when things are observed or not observed for various reasons. And so longitudinal data can be kind of tricky to work with, and our kind of most common statistical methods aren't always kind of up to the nuance of what's going on. And so what I want to describe is kind of the underpinnings of what we call multi-state transition models. And the reality is that an individual has some kind of underlying continuous time transition history. Of course, there's not really a specific cutoff between one day that one has normal cognition and one is impaired, but there's a point at which we could say someone would kind of diagnose this had they observed them on a given day. And so we can imagine that an individual comes to their first visit, they have normal cognition, they're impaired but not MCI for the next two. Some point between visits 3 and 4, they pass through MCI and all the way to dementia. And so that's one possible trajectory, but the specific times we don't observe. We only observe data at specific observation times, these different visits. And so the only information that we have is not when these transitions occurred, but that they've had normal cognition at visit 1, impaired but not MCI at 2 and 3, and dementia at visit 4. And so multi-state transition models, which I work with quite frequently, are one approach to estimating what we call the underlying continuous transmission rates. And so it's kind of like trying to understand the kind of pressure to transition or the intensity to transition. It's not quite likelihood because these kind of come together to form likelihoods, but I have examples in the next couple of slides. So in this case, it might be that there's not an individual who has normal cognition, any given year has not that much of kind of intensity of becoming impaired, but once you kind of start the process, each subsequent transition becomes more likely. A little bit of kind of behind the scene details here. Continuous time stochastic models. So multi-state transition models are continuous time stochastic models. They're tracking a person. Sorry that I reused the slide. It says tobacco use here, but in this case, it's kind of cognitive status. The transitions can occur at any time, but we observe only the state at certain times. And that we typically take kind of a Markov approach here. So the transition probabilities depend only on the current state and not on one's past history, although we can account for past history through covariates. And so what the multi-state model is doing is taking multiple competing possibilities into account when estimating underlying continuous transition rates. So we might say that in one current state that there's 3 possible future states, and given some number of individuals, we can see that only about 12% of people remain in their current state, and about 2% go to the first, 53 go to the second, and 33% go to the third. And so given this, we can, this tells us about the probability of remaining, tells us about that kind of overall pressure to transition. And each of the distributed probabilities tell us about how you divvy up that overall transition rate or transition hazard into possible future states. And this is fairly straightforward. So we can assign some numbers to those things given these numbers. But if you have a fairly complicated or interconnected network of states, this becomes a little bit more complicated because you have lots of potential competing transitions at work here. In the context of cognition, we're looking a little bit more at like a chain of probably like forward and backwards and not necessarily jumping there, because you kind of have to go through intermediate states. However, we might also be interested in changes in the predictors, so that we might be interested in different possible biomarker states or different possible neuroimaging states, and so how those transition across time might be more complicated. So I've worked with these models in a lot of different applications in the past, primarily in tobacco control. So thinking about how different product use people transition. A lot of this kind of comes from the questioning the harm reduction perspective of whether e-cigarettes help people quit cigarette use. Are they preventing people from quitting, and then kind of answering those questions, and how that's changed over time with different generations of e-cigarettes. I've used this kind of in an infectious disease dynamics world, looking at how individuals become HPV positive and kind of clearance of that human papillomavirus, which is a teleological agent of certainly cervical cancer, but other types of anogenital and oral head and neck cancers. I've also been advising a colleague on the use of these models in chronic obstructive pulmonary disorder. And so these are different states of COPD, and looking at predictors of progression versus regression in COPD disease. And I'll have some plans with some other folks to look at kind of cardiovascular disease. So kind of wide applications, and I'm looking forward to doing it here with Alzheimer's. So what we get out of these models, the hazard rates themselves are useful, but tend not to lend themselves to direct interpretation. So I kind of want to give you a taste of the sort of things that we're going to be able to get out of these models. So these heat maps, I think, are kind of a convenient way of visualizing the cumulative transition probabilities. So if we look across a row, we can see the probability of transitioning to each state within kind of one time point, so maybe 1 year. This is an example from tobacco control. So if I look at people that are kind of exclusive cigarette users here, annotate, then we can see that about 85% of exclusive cigarette users are likely to continue to be exclusive cigarette users after 1 year. Almost 10% of them may have not used any product in the last month. So kind of non-current, maybe not necessarily have quit completely, but are not currently using. And some small fraction may have switched to e-cigarettes or e-cigarettes and cigarettes. And so we can, this sort of thing we can imagine for progression along sort of cognition pathways as well. And you might be saying like, okay, this is interesting enough, but basically we could do this just by kind of looking at the number of places where people go. Well, what we can't do, so let's annotate here, just looking at probabilities is looking at hazard ratios. So trying to understand how different characteristics or kind of testing results may be more or less associated with kind of faster transition rates. And so this is an example, again, from tobacco control, looking at current exclusive cigarette users and whether they are likely to pick up e-cigarettes and become dual users. So in this example, we can see that non-Hispanic Black and Hispanic exclusive cigarette users are less likely to transition to dual use than non-Hispanic White users, and that we have this lovely gradient here as the younger folks are more likely to pick up e-cigarettes to become dual users, and older users are less likely. So it's very consistent with what we would expect based on patterns by age. And so what we can imagine seeing as a result from my proposed project is that, you know, for different cognitive testing or biomarkers or imaging, we can say that for each of these different transitions, how much greater or reduced is the rate of transitioning to the next state. So kind of get to the specific aims. So the aims of my pilot project are to, one, develop a multi-state transition model of Alzheimer's disease progression, and then leveraging the data in the Michigan Alzheimer's Disease Research Center cohort, we're going to estimate associations of progression rates with sociodemographic, biomarker, neuropsychiatric tests, neuroimaging, and other covariates. Where this is going is that I hope to kind of leverage the work that we do with this cohort to support external funding application to apply these methods to the larger NAC data set, which has about maybe 40,000, I think, people compared to, I think currently about 800 in the Michigan. We expect to kind of in the future include more specific dementia outcomes or different types of dementia, which can really only be done on a larger data set. And I think the thing that I'm really looking forward to kind of in the big picture is to develop an integrated multi-state model to simulate individuals' future cognition covariate values, both from this perspective of personalized forecasting with uncertainty, but also population level predictions. And so that's kind of the big picture. What do I need to get there? I think, as you may have recognized, that I'm not experienced with Alzheimer's and dementia. And so what I'm looking forward to is this curriculum to kind of learn a bit more about kind of what I need to know. Looking forward to working with my mentorship team to kind of discuss appropriate model covariates and model interpretation. The data team has provided me with a kind of data dictionary, and as someone that hasn't worked, I'm kind of like, I don't know which of these variables is appropriate. So Kelly and Nick and Jem, looking forward to working with you. And since I'm now on the tenure track, grant writing is a kind of a big push for me. So I'm hoping to get grant writing feedback as I pursue NIH sponsorship. I think that's all I have for today. So I guess I'll stop here to see if there are questions. Things in the chat here. Does the data set include measures of physical activity? Maybe that's a question for John and Kelly. I assume some amount, but I don't actually know off the top of my head.
Yeah, I think from what I remember, that the physical activity measures are a little bit crude. Some of the sites have specialized in that. For example, our former data core colleague Hiroko Dodge has at Oregon Health and Sciences University included Fitbit measures of physical activity, and those have been really helpful for their group. So yeah, I think that's a great point, and we might be able to consider physical activity at different levels of specificity depending on the data source.
Yeah, that's a great suggestion as a kind of another piece of the puzzle. Thanks.
Okay, very good. Andrew, thanks very much. Why don't we go on to our next talk? I think Liz Litkowski is next. Liz, do you want to introduce yourself and then go ahead, please?
Well, I need to unmute first. Yes, thank you. Maybe I swap slides here. Right, I think you're seeing my whole presentation, right?
Not yet.
Oh, you're not. Oh, forgot to share.
Okay.
How's that?
All right.
Great, thank you. Okay. Yes. Hi, I'm Liz Litkowski. I am a genetic epidemiologist by training. Excuse me. And today I'd like to talk to you and share results from, or share my work of genetics and diet and how sex differences explain a relationship between glucose metabolism and cognition. So my background is a little bit different than a lot of folks. I am not from academics originally. I'm actually from industry. And I spent some time as a hardcore computer programmer of image processing data from satellites, and then moved on to do a little bit of work in quality control throughout companies. both in the high-tech industry as well as the organization that I worked for. I then moved to the oil and gas industry for a little while, looking at intricate royalty payment systems, as well as drilling engineering processes in that industry. And then I decided to go to the good side and joined epidemiology at the height of the COVID pandemic. And my area of interest is related to diabetes and dementia. Both of these diseases have very high prevalence in the United States. And we know that the diseases differ greatly by different ancestries, whether that's due to genetics or whether that's due to social environment. We also know that there's a strong relationship between diabetes and dementia, that there's a very high risk of dementia in those who have been diagnosed with diabetes. And yet there is some evidence in recent history that some of the treatment and or prevention of diabetes has actually shown a decline in dementia in some cases. So given all of the observational evidence of the relationship between diabetes and dementia, I was interested in pursuing, is there some kind of sharing between these two diseases that would help explain this relationship? In particular, my work in the Million Veteran Program, I was able to demonstrate that there was indeed a shared genetic association between diabetes and dementia. And in particular, because the Million Veteran Program has a fairly diverse population, I was able to see this in both non-Hispanic white individuals as well as non-Hispanic Black individuals. So I was able to see the genetic components of that with this first study that was published in Diabetes Care. And then secondly, I worked on Mendelian randomization in the same population to look for evidence of causality between these two diseases. I was able to establish some evidence of causality, both in the non-Hispanic white as well as the non-Hispanic Black population. So for those of you who don't know Mendelian randomization, it's a technique typically used in human studies because it's, well, mostly because of ethical reasons that you can't conduct clinical trials in some cases to test what you really want to test in humans. In particular, here is the evidence that we found within this population. In the non-Hispanic white population, you'll see that for higher and higher elements of genetic risk of diabetes, the odds of dementia are increased. So you'll see a trend line that moves across the different quartiles of genetic risk. And this was observed, again, both in non-Hispanic white individuals as well as non-Hispanic Black individuals. And in particular, vascular dementia, which you'll see here, has a higher effect size. The effect size that we observed in the non-Hispanic Black population was actually greater than it was in the non-Hispanic white population. But this, of course, left me with just wanting to know why. Why does this happen? Why is there a relationship with these two diseases? And so I started just perusing the literature to find out more information. A really good source of this information was this review article that I found by the Chavoya in 2019, which tried to segregate the different elements of potential causality. Impaired insulin signaling and transport, which is my particular area of interest, but then also what other metabolic changes are caused by type 2 diabetes in terms of inflammatory state, as well as some of the genetic factors like mitochondrial dysfunction and the ApoE genotype, but also structural neurological changes. So just looking at brain images, it's not clear whether the alterations that we see in those images are caused by diabetes or whether they're an after effect of some of the dementia diagnoses. So again, still more curiosity. I feel like a little kid sometimes, but but what is that? So, so that brought me to the Kazarowski lab. Let me see if I can- in earlier this year in 2023, I moved to the Kazarowski lab to take advantage of an opportunity to look at this in more detail, to really pursue cause. And so the mouse panel, the ADVXD mouse panel, which for those not familiar with this, the ADVXD line is built on several well-established lines. So the AD model, which is known to manifest amyloid accumulation as well as cognitive deficits over time. Separately, the BXD panel, which is derived originally from a B6 and D2 mouse panel. These, the BXD panel is formed based on multiple generations of recombinant inbreeding with these two lines, and that's where the diversity of the mouse panel arises. So at this point in time, the F1 hybrids will segregate for approximately 5 million sequence variants. So something that is not quite at the level of humans, but at least it gets closer than just having a few variants that are different. So the other advantage of the ADVXD line is that the hybrids are fully isogenic, which means that if we find a particular strain that has a particular susceptibility or resilience to specific conditions, we're able to then rebreed that strain in order to do more isolated testing and more specialized testing for those particular strains. So we conducted a controlled study to look at mechanism. We looked at 39 specific genetic strains from the ADVXD panel, and we used varied conditions for those the mice that participated in our study. 2 different diets: a high-fat, high-sucrose diet, which is also commonly referred to as a Western diet, or a 6% extruded or chow, which is considered to be a healthy diet. 2 different genotype types, AD or non-AD, and then 2 sexes, of course, that we're interested in. And so this forms to 8 different groups so that each group has a combination of those particular conditions: female, high fat, high sugar, with the AD genotype, the same thing, but with a non-AD genotype, etc. And so then we're able to study what kind of results do we have with respect to cognition when we have put these mice through the paces of each of these different conditional environments. So a couple specific things that we were looking at to test glucose metabolism and memory. So in particular, we used a glucose tolerance test in which fasted mice receive a glucose injection, and then glucose clearance is measured over several intervals for a 2-hour period of time. And in particular, in the study that I'm going to talk about today, we measured peak glucose under multiple conditions. So my toy example here is just showing peak glucose as measured under 2 different conditions, but we were doing all of the combinations that I had previously mentioned. Then with respect to memory, we conducted contextual fear conditioning in which mice are moved to a novel environment. They receive a shock in that novel environment. They're then returned to their home cages and then returned to the novel environment the next day. And in the second day, they are not exposed to that shock. But we then measure how often they freeze in that new environment, which is a measure of memory. Do they remember what occurred the previous day, even though they are not experiencing that on the second day? Both of these measures are taken at 6 and 14 months so that we can look at not only what happens at particular ages, but also look at changes over time. What we did observe in this study was that an increase in that peak glucose measure in terms of the change from 6 to 14 months, an increase in glucose and peak glucose is associated with a decline in memory over that same time frame. This is for females with the AD genotype on a high-fat, high-sugar diet, and about 21% of the variance is explained with this model, and it's got a statistical significant value. However, under the same conditions, AD males on a high-fat, high-sugar diet do not. For this group, we did not observe the same kind of relationship. I know this line looks a little bit like it's going up instead of down, but basically it's flat given the P value that we observed. And let me just point out that an increase in peak glucose over this time frame means that their glucose metabolism has actually declined. So, oops. Just wanted to make sure that wasn't a question for me. So from this model, we actually now have a couple of aims that we'd like to pursue. For aim 1, we would like to take advantage of access to an Internet Interventions Testing Program, ITP, for diabetes drugs, because there is evidence that diabetes drugs may have an effect. We can start a new experiment in which we have several different treatment groups, a control group, but 3 different potential drugs, rapamycin, erythritol, and canagliflozin, and we can test them on specific susceptible or resilient genes. Remember, I had that declining line that showed the decline in cognition over time. We're interested in looking at not just the strains that exhibited poor glucose metabolism, but also the ones that were resilient under in that environment. So a few of the strains that we'll be looking at are ones that are resilient as well as susceptible. And however, in addition, we've got genetic information to be able to get closer to the mechanism. And so as long as we've got heritability assessments that are reasonably high, we can do things like quantitative trait mapping and look for specific genes of interest. So let me show you a few of the preliminary results that we've seen from that. We have indeed found that there is a high heritability for both peak glucose change and memory change over this time frame. And so that makes it a good candidate to look at genetic mapping. We have observed that the genetic disposition is polygenic. So you'll see this is across the chromosomes within the VXD population. We are able to see peaks within this genome range. And in particular, in chromosome 9, we're able to see a particularly steeper peak. We did do a few tests just to make sure that we understood how each of the different covariates affect the outcome that we see between glucose metabolism and cognition. And we were able to observe that male sex and a non-AD genotype do attenuate the decline, but a high-fat, high-sugar diet increases that decline. That last we would expect. And so we do now have a set of genes to take a look at. This needs a lot more work at this stage because we have identified the gene region and a few genes that have been identified in that area. Interestingly enough, we see some expression levels associated with the central nervous system. We also see some cellular changes, particularly in cells that are related to the nervous system, as well as some development of the nervous system itself. This requires a lot more work, so stay tuned for future results. But in conclusion, the key thing is that we found that the ADBXD panel does parallel human findings. Because we know that there's so much diversity in our human studies, we want to be able to do something that's translational by using a model that is also very, very diverse. We've seen that both glucose response and cognition are related to heritable traits, that we can modify the effects by sex and diet, and that the trait is polygenic, but that there is a particular area of the genome that will be interesting to investigate. So that is, that's all I have on my own topic, but also just wanted to thank the collaborators in the Catherine Kazarowski lab, which keeps growing day by day. I've also had significant contribution, in particular from Dave Bridges, who was in the School of Public Health, who has also assisted in my award in the 232 for basic diabetes research as part of that. And we did paint the rock as part of our celebration of the 10-year anniversary with Kazarowski. So just had to throw that one. That is all I've got.
Yeah, there's a question in the chat, Liz.
Yes, I see. Okay. So yes, so the diabetes treatments, the research that I've read has been done more at population levels, so just general studies, potentially aggressively treating diabetes. I don't know, that's part of what I'd like to study with the interventions test protocol. So I am hoping that we will be able to learn some more as part of this. I do not have a specific accounting for physical activity in the work that I did in human genetics. That is data that we capture as part of our mouse studies. And so it can be an element of that. There is a lot of evidence about increase in exercise being related very closely with insulin resistance versus insulin sensitivity in muscles in particular. And so that is actually something that is very, very interesting to me. Thanks for the questions.
Well, just a comment about that. So there have been a couple of studies of intensive therapy for type 2 diabetes with looking at cognitive measures as an outcome. In fact, actually the effects were negative. The more aggressively treated group actually had some worse decline, further decline in cognition on relevant measures. So it's a little uncertain at this point. So the relationship,
right.
Yeah.
Take the there's a delicate balance.
Yeah. So it may be, in fact, that aggressive treatment actually produces some relative hypoglycemia, which may be a bad thing. So actually, Liz, a question for you. Have you any of the genes you found out have anything to do with endothelial function?
So not in what I have found so far. My literature review has actually pointed to an area that's really interesting to me in terms of glucose transporters. So typically related to endothelials. I think it's GLUT1 that is the transporter from the endothelial cells to astrocytes. And then from astrocytes, there's several other glucose transporters that are involved in then transmitting to neuronal cells. And so that's more what I have found from literature research, but it looked really interesting to me. But so far, my individual study has not pointed me in that direction. But I am really interested in the tests that demonstrate that.
Very good. Any other questions? All right. Let's go on to our third talk. I think that's Norman Scheel from MSU. Norman, would you like to go ahead, please?
Yep. I'm sharing my screen. Can you see that?
Not yet. There we go.
Okay, welcome to my talk from your friendly neighborhood Spartan. So first I'm going to start with a short introduction, then talk about my current main projects, and then talking about complexity of the brain as in our in my MA-DRC project. So first of all, I did my Master's of Science in Informatics in 2011 in Germany at the University of Lübeck in computer science and medical informatics and medicine. So if you've never been, Lübeck is a very amazing city at the Baltic Sea in the north of Germany with a lot of history. My house back there was built in 1680. It was quite the adventure to live there. So if you ever get the chance, go to the north of Germany. It's very, very beautiful. Then in 2017, I continued in Lübeck to do my PhD in machine learning and computational neuroscience at the Institute for Neuro and Bioinformatics. And there I mainly looked at resting state fMRI. And basically, I looked at all the different modalities you can calculate out of the resting state fMRI. But everything first starts with pre-processing, data cleaning, and the different noises in the signal. So that's basically where I spent most of the time trying to understand all the different data components that are in resting state fMRI that can have an influence on all the higher level things we compute out of them. And then I used the connectomes to do predictions of genders, Parkinson's disease state, and ages. I did classification and regression using support vector machines, and I specifically looked at high dimension, low sample size problems there. Then there are the resting state networks that everyone is talking about, and specifically the default mode network was very interesting to me as it shows these anti-correlations to the other networks. And I looked at phase synchronizations between the different networks. And then went on to temporal functional modes, which is kind of an extension to resting state networks. Again, looked at reproducibility because they were kind of new. And phase synchronization, I looked at probabilistic functional modes and QPPs, which are quasi-periodic patterns that you can find in a resting state fMRI. All of this kind of looks at the fourth dimension. Speaking of time, what does the time component tell us about cognitive states? And then the last part I looked at was complexity of brain function as it's regarding to age. I looked at it in local parts of the brain or in global overall, or in a searchlight or small windowed approaches. Now, during the time in the early 2010s, there was a big paradigm shift in fMRI, as typically before, when we recorded a resting state fMRI, we did so in an alternating slice, but one slice at a time. All of a sudden, we had a new sequence called multiband imaging, where we can all of a sudden record multiple slices at the same time. So we can image a lot faster, but again, circling back to the pre-processing and data cleaning and noises, that also meant that there's lots of other things to consider, again, with the respect to all the outcomes. So that's also where I spent lots of time analyzing. So in 2018, I came to Michigan State University to the lab of David Zhu here. And with him together, I'm the co-lead for our imaging core for multiple clinical trials. The main trial that we just concluded imaging was the RAD, risk reduction for Alzheimer's disease. And that one looks at the question, what's good for the heart is good for the brain. In this grant, we're testing strategies that may reduce the risk of Alzheimer's disease in individuals that are at high risk. So their age range is 60 to 85 years. they have hypertension, they have a family history of dementia by a sibling or a parent, or they have some concerns about their memories. We divided these people up into 4 arms of intervention. One was rigorous aerobic exercise. The second one was intensive medical management of blood pressure and cholesterol. The third one was the combination of exercise and medical management. And then the fourth one was the standard care. We have 4 testing sites with 5 3T MRIs of all manufacturers. They're located at the University of Texas, then the University of Kansas, Louisiana State University, Washington University. Yeah, those were the 4 sites. And we try to harmonize the different sequence according to the ADNI 2 protocol. So we started with a high resolution T1 MP rage. We had functional MRI. We have ASL for perfusion. We have diffusion MRI for structure. We looked at FLAIR images for the white matter hyperintensities, and we have PC2D images at the neck to measure the flow of blood into the brain and out of the brain. And so the first thing that I did is when you want to do like more high resolution comparison between subjects, you all have to register them into a common space. And typically this is done using the MNI 152, which is based on 152 20-something-year-old college students. And as you can imagine, these brains of these young, healthy students look very different to the brains that are in our cohort. So I developed this RAD 420, which is based on 420 subjects that had really high quality scans. And I developed this standard brain that is applicable for our cohort, but can also be extrapolated to other cohorts in the same age range. Because you can see like there's a lot more atrophy going on. The ventricles are a lot bigger. So that the artifacts that you get when you stretch an older subject into a common space are not as big for this template as they would be if we would stretch them to fit MNI 152. And then I extended that to all the different modalities of FLAIR, CBF, diffusion MRI, and created functional atlases, and still working on the structural atlas here. That was basically the RAD project, and since that just finished, we had a small preliminary study where we looked at amyloid uptake from PET imaging, and we compared that to the fluctuation in bold fMRI. And we could find, especially in the posterior regions, there is a correlation between bold fluctuation and amyloid uptake. So that was the basis for our new grant that just started, which is called IPAD, the impact of intensive treatment of systolic blood pressure on brain perfusion, amyloid, and tau. So in these subjects, again, we have high intervention on the blood pressure, and then we're looking at over a span of 2 years, how that changes the uptake of amyloid beta and tau through PET images. And now let's come to my current MADRC projects, which is a development project under the REC mentorship project. So my mentors are Kelly Bakulski, Jessica Damoiseau, and Dave Morgan. And let's start very basic with MRI for the non-imaging people in the group. So first of all, the basis of every scan is an anatomical MRI scan, a T1 scan, where we typically have a spatial resolution of 1 by 1 by 1 millimeters for each voxel. So it takes a couple of minutes, roughly 5 to 7 minutes, to take the whole volume and get the anatomical details. The other modality that I was talking about was the functional MRI, where we look at how the brain is working, basically. We have a typical spatial resolution of 3 isotropic millimeters and a TR, so a repetitive time of 2.5 seconds, where we take a brain volume every 2.5 seconds. Now with the multiband, as I mentioned earlier, we can do that a lot faster now. And we're saying functional MRI and we look at brain activation, but we cannot directly image a neuronal function per se. We're looking at the BOLD effect, which looks at different oxygenation status based on the metabolism of neurons. So it's not direct a brain function, but a correlate of it. And in the resting state fMRI, we say we don't have a task, and we just say, let your mind wander freely. Don't sleep, don't fall asleep. Yeah, whether they have their eyes open or closed is different, and it's a whole different discussion I'm not going to go into. So when we take this functional MRI and we just project that onto the brain and put a little bit color to it, then it kind of looks like this. And we see there are some organizations there, but it's really hard to grasp. And so as a data scientist, I try to look at this in a more data science perspective. So we have a brain that's made up of all these voxels, and we just line them up on the strings, so as a vector. And in the functional MRI, we just have that many of them. So we can create a matrix that is space by time. So basically saying the voxels over time. And a very standard algorithm a lot of people use to decompose this data is the independent component analysis, where we divide the data by spatial independence. And so we end up with spatial components, and each component has a time course. If we do that to the data, so this is again our brain activity, we get all of a sudden all these different networks, and we look at how these networks are coming in and coming out and are working with each other, which is kind of interesting. And it turns out this is highly reproducible. So there have been many publications, like even an early one of Jessica Demoiseau, one of my rec mentors, showing that these are highly reproducible in a lot of subjects. Now, when looking at the complexity, actually a first step to do this independent component analysis is the principal component analysis, where we look at the distribution of the data along the axis of the variance. And here for a 2D example, we have the principal component, first principal component is along the direction of the highest variance. So this gives us the first principal component with the eigenvalue lambda1 showing the strength of variance in that direction. And the second principal component is the second dimension of variance. And so this shows us the extent in that direction, giving us the second eigenvalue. Now, for fMRI data, we have a lot more dimensions, but this is how you can think of it in a low dimensional space. In 1996, there was a publication by Jiří Vackerman from the Czech Republic, who looked at how to quantify complexity of brain function in EEG, and he developed the omega entropy, basically taking the eigenvalue spectrum and calculating the entropy of it. And that is followed by, in 2012, colleagues of mine in Lübeck, Germany, they developed the multivariate principal subspace entropy, crazy complicated name, but it's actually not that complicated. Using this entropy, they were able to show in a functional MRI task paradigm that the complexity changes with task onset and task offset. Now, when we looked at that, we found, I forgot to mention, the cool thing about MPSE versus omega is that we have a parameter K. So that means we can look at sub-portions of the eigenvalue spectrum, while omega just goes from I to D, meaning it uses the whole eigenvalue spectrum. And with this parameter K in MPSE, we can limit the eigenvalue spectrum to certain parts of it. The problem is that the eigenvalues in MPSE are directly taken from the principal component analysis, while the eigenvalues in omega are normalized to a unit sum. Basically, you sum them all up, divide them by the average, so you end up with a normalized eigenvalue. And so we found out this causes MPSE to be highly dominated also by data variance in itself. And this is why I then created this normalized MPSE, where basically the only thing we changed is we normalized the eigenvalues to a unit sum now. And with some simulations, we could show, okay, if we have a increasing variance, MPSE is also dependent on the variance, while omega and the normalized MPSE both are independent of it. Now, to look at that in actual data, I used the Rockland sample from the Nathan Klein Institute, where we have 186 subjects from 18 years to 85 years. And we excluded subjects that had more than 3 millimeters of motion. We used the multi-band resting state fMRI sequence with 2 by 2 by 2 millimeter voxels, a TR of 1.4 seconds and 404 volumes, making up 9 minutes of scanning time. And we could show that actually the complexity is different between older and younger subjects, meaning older subjects had a high entropy, meaning a low complexity, and younger subjects had a low entropy, meaning a high complexity. And this, we found this significant correlation for that. Going into more detail, as I said, the cool thing about MPSE is that we can look at certain amounts of eigenvalues in comparison to omega that uses the whole eigenvalue spectrum. So looking at the whole brain complexity age correlation, omega can significantly differentiate the ages between the subjects. But when we look at here for MPSE, if we just give it like 50% of the eigenvalue spectrum, then it's not very significant. But the more data we add, the higher significance it becomes. And you can see with K76, meaning it reaches 99% of eigenvalue energy at 76 components of 403 possible components. So that means the first components carry most of the information in this data set. And if we look at the different networks, we can see except the higher visual, every network was able to significantly predict the ages, but at varying levels of number of components, we need to do the prediction. So the sensory motor, for instance, needs the lowest amount of components to do a significant age prediction. Then auditory, which we think might be some compensatory mechanism. Interestingly, the default mode network is kind of in the middle, but overall, what we can take out of it, different networks need different amounts of components to do the age prediction. Now, when we look at the searchlight approach, we can actually pinpoint in the brain where the complexity changes due to aging. And with K equals 76, meaning we have 99% of eigenvalue energy, we can see quite a lot of components are significantly changed between for aging, and especially the motor area is bringing into my mind, and also the posterior cingulate, which is part of the default mode network, which was quite interesting. Now, if we lower this to K6, meaning only using the first 6 dimensions, which include 50% of the eigenvalue energy, the motor areas are like the most discerning factors here for aging in this cohort. So if we put this together, we can find 70% of energy is distributed across the first 20 components in the data, which is really nice because typically when we do the full ICA, we find about 14 to 21 resting state networks altogether. So there is this nice correspondence between the principal components and the independent components in our data set. Then 50%, more than 50% of eigenenergy spans across the first 10 components, meaning like most of the information is right at the front. And for the whole brain, the information relevant to decoding aging is found across a range of components, and it's not dependent on a single component. So we can see it's multiple networks working with each other where we can find the difference. But for a single regions, we need different amounts of energy in order to do the prediction. And the regions that only need a few eigenvalues to do the prediction, those are highly susceptible to aging here in this case. And we can use K as a filter to sort exactly these regions where we know there is a strong correspondence to aging. And this leads to the first research questions and aims in my development project, where we ask, can fMRI dimensional complexity measures to be used to predict AD biomarkers? So we want to investigate mechanisms of cognitive decline and aging through changes in functional complexity. And we want to evaluate functional complexity for detecting and predicting AD and dementia. And we want to take advantage of the MADRC longitudinal cohort and investigate the longitudinal changes in fMRI dimensional complexity, because this complexity measure has never been evaluated on any bigger data set except the one that I showed. So this will be very interesting if we can use that as a precursor to that. And now we switch gear from functional MRI to diffusion MRI. Just as a real quick introduction, in diffusion MRI, we switch on gradients in many different directions really fast. So we can look at how water is diffused along different directions. So when it hits a wall, water cannot diffuse in that direction. So that's how we look at how fibers in the white matter are actually traversing the brain. So you can see if in this direction, the corpus callosum back there is not showing, and here you just see half of it. And if the direction just comes right out of the screen, you can see there's a full display here. So now we do that with a lot of directions very, very fast. And what you can get then is for each voxel in the white matter, again, you can do a principal component analysis of the directionality of the water diffusion. And actually, this has been done in the pixel-based analysis of the diffusion images. And basically, it's mathematically the same concept that some voxels have high complexity, others have a low complexity due to water diffusion. And this has been shown. So Andrew Bender, who is also in the imaging core, we talked a lot about this, and that raised the question if there is a correspondence between the complexity of the fiber distributions in the white matter and the complexity of brain function itself. So this is the first question. And then if we can combine these measures to even have better predictive value of MCI and AD outcomes or prognosis. And with that, are there questions?
Okay, Norman, I think there was one question in the chat.
Oh, okay. Let me see. Where can I find it? Oh. Okay, so first, how are you constructing the networks? Yeah, let's go back to this slide. So this is basically the independent component analysis, what I talked about. This is basically, this is the input. After all the pre-processing of the fMRI data, we end up with this space times time matrix. And then we do independent component analysis on it, which divides our data into spatial maps and time courses. So each spatial map then has a time course, and a spatial map then is a network. Now we end up with a lot more spatial maps and time courses due to noise in the data, but typically we find about 14 to 21 of these spatial maps are the corresponding networks. There are other ways how you can construct networks through seed-based connectivity, where you put a seed in a certain region to elicit certain networks. But the data-driven approach is using independent component analysis because you don't have to put any priors into it.
I guess my question was really around when you're giving a name to the individual networks. I wasn't sure how that relationship was established.
When you what? I didn't hear.
That you were giving a name to each of the individual networks that you found. I feel like it was maybe an earlier slide than this.
You mean like this one?
Yes. That.
Yes.
How did you?
So basically, I mean, it's really difficult to tell from this from these images here. But if you look at the maps directly, through the axial slices in a 3D viewer, you can basically look at the brain regions that are activated in this, and so that's how you tell. Okay, this is default mode activity. This is the visual cortex, so this is the visual network, and that's how we discern these different networks.
Okay, great, thank you.
All right, and if a participant was not supposed to fall asleep in the fMRI, but they did, was the fMRI repeated? You know, that's a very tough question because you cannot, I mean, I'm working with Katie Cheng from Vanderbilt on this a little bit because they do concurrent EEG and fMRI. And through the EEG, you basically can tell if there are sleep spindles forming. And that's how you can tell if people actually fall asleep. Some paradigms tell the subject to close their eyes, but don't fall asleep, which is really difficult to monitor from the outside. If you have, like some others have a fixation cross that you're supposed to watch, there you can have like an eye tracker to see if they close their eyes and fall asleep. You could do maybe a little bit different prediction there, but having the eyes open completely changes the activation patterns in your brain because the visual network is a lot more activated because your eyes are open. So falling asleep detecting is really difficult, and there are correspondence with the global signal. The more drowsy people are, the higher, and yeah. Or you can, of course, you can ask the subject if they fell asleep, but that's really difficult if they can even remember and if they would be truthful. Some people in the early stages, like in the early 2000s, people had caffeine drops in the waiting room so that they would use caffeine to make them more alert, which also was helping getting better signals. But I mean, of course, that's not practice anymore, and you cannot just do it like that. And yeah, it's difficult. Falling asleep during resting state is a difficult problem that we're still trying to solve.
All right, thank you, Norman. I think Dr. Torres, you're next, actually. So.
And actually, Stephanie has slides,
Okay.
not just slides, I'm sorry. I actually I gave her a video because there's a lot of noise in my background. So Stephanie, if you could play my video, that would be great.
Hi everyone, my name is Anneliese Torres, and the title of my presentation is Physical Activity Across the Life Course, Biological Aging, and Risk for Alzheimer's Disease and Related Dementias. This is the outline of my talk. I'm going to start by introducing myself, then the proposal. I earned my PhD in nursing from the University of Michigan in Ann Arbor. My dissertation was focused on the association between physical activity and depressive symptoms in Black U.S. adults. During my postdoctoral fellowship at the University of Iowa, I examined the association between physical activity and areas of the brain related to depression. As junior faculty at the University of Wisconsin, I compared the effects of physical activity with mindfulness-based stress reduction in a randomized controlled trial. In my current position, I'm examining the association between physical activity and areas of the brain related to Alzheimer's disease and related dementias. Alzheimer's disease and related dementias, or ADRD, have been associated with shorter telomeres indicative of advanced cellular aging. Physical activity has been associated with attenuated ADRD pathology, preserved cognition, and preserved telomere length. Telomere length is more useful in assessing biological age when used with other markers, such as epigenetic clocks. It is not known if telomere lengths and epigenetic clocks are mechanisms by which physical activity attenuates ADRD pathology and preserves cognition. Knowing the mechanisms by which physical activity attenuates ADR pathology and preserves cognition is expected to reduce the risk of ADRD. As there is no cure, there is a critical need to reduce the risk of ADRD. Moving on to my objective. The long-term goal of my program of research is to determine the sustained effects of physical activity on the brain. The overall objective of this proposal is to determine a mechanism by which physical activity across the life course attenuates ADRD pathology and preserves cognition. The central hypothesis in this proposal is that biological age is a mechanism by which physical activity across the life course is associated with less amyloid burden and preserved cognition. The rationale for examining physical activity across the life course is that ADRD prevention may be most effective when administered 15 to 25 years or more before expected ADRD symptom onset, that is, when ADRD pathology first appears in the brain. This project leverages existing data collected in a prospectively followed cohort of middle-aged and older adults. There are 4 specific aims. The first is to determine the association between physical activity across the life course and amyloid burden. The second is to determine the association between physical activity across the life course and cognition. The third is to determine the association between physical activity across the life course and biological aging, specifically telomere length and epigenetic clocks. The fourth is to determine if biological aging is a mechanism by which physical activity across the life course is associated with less amyloid burden and preserved cognition. The goal of my training program is to gain experience in cognition in biological aging. I am somewhat familiar with amyloid. However, I have limited background in cognition and biological aging. I will complete an online curriculum about contemporary dementia research to get introduced to basic features of dementias. I will meet with one of my mentors 1 hour monthly to discuss cognition and the varied methods of measuring cognition, i.e. memory, executive function, and a global composite score. I will meet with another one of my mentors 1 hour monthly after the biological clock data are available to discuss results. I will attend mentoring committee meetings every 6 months. I will participate in the Career Development Workshop jointly sponsored by the Michigan ADRC and the University of Michigan Pepper Center in the spring, including a mock study section and other career development activities. I will present findings at the annual Michigan ADRC Symposium and National Alzheimer's Disease Research Center meetings, publish results, and submit an R1. Participants come from Visit 1 or baseline of Dr. Benlin's study, which was part of the Wisconsin Registry for Alzheimer's Prevention study in the Wisconsin Alzheimer's Disease Research Center. They were not to underwent a comprehensive cognitive evaluation, neuroimaging, whole blood collection, and assessment of lifetime physical activity at baseline. Participants were middle-aged and older adults with no cognitive symptomatology at baseline, and the sample was enriched for participants with parental history of Alzheimer's disease. Exclusion criteria are known family autosomal dominant mutations, MRI scanner incompatibility, history of major psychiatric disease except depression, major medical conditions, specifically a history of neurologic disorders including prior head trauma with loss of consciousness, cancer requiring chemotherapy or radiation, insulin-dependent diabetes, and untreated hypertension, or abnormal structural MRI indicating disease or a condition that would affect normalization procedures such as a tumor or hydrocephalus. Participants' consent was obtained under a protocol approved by the University of Wisconsin Health Sciences Institutional Review Board. The IRB at Wayne State determined that this proposal is not human participant research due to de-identified private information in human biological specimens. Therefore, IRB review was not required. Physical activity was measured with face-to-face interviews with participants at baseline using the Lifetime Total Physical Activity Questionnaire, which measures 4 domains of physical activity: occupation, transportation, household, and leisure time. Physical activity is measured across the life course with cutoffs chosen based on Erickson's theory of psychosocial development. The stages relevant to the current application include school age, adolescence, young, and middle adult. The hours per day, days per week, weeks per month, months per year, and number of years of each activity within each domain was recorded. Metabolic equivalent, or MET, is defined as the ratio of the metabolic rate from activity to the resting metabolic rate, was abstracted from the compendium of physical activities and assigned to each reported activity. This is a standard measure used in physical activity research. The average MET hours per week per year spent in each activity from kindergarten through middle adult were recorded. Physical activity was categorized as sedentary, light, moderate, and vigorous within each domain, within each developmental stage. Evidence of validity was only found for moderate and vigorous intensity, which is used in this proposal. Cognitive testing results at baseline will be used. Cognitive testing assesses general performance, IQ, memory, executive function, and processing speed. Cognition was measured with the National Alzheimer's Coordinating Center Uniform Dataset Battery, Gray Auditory Verbal Learning Test, Controlled Oral Word Association Test, and Trails Making Test A and B. Memory, executive function, and a global composite similar to that used in other studies of preclinical Alzheimer's will be used as primary outcomes. Amyloid deposition was measured with PET imaging. 8 regions of interest, as you can see here. For telomere length, DNA will be extracted from whole blood obtained at baseline. DNA will be assayed for leukocyte telomere length using a validated quantitative PCR method to determine mean telomere length across all chromosomes, as previously described by Dr. Charlotte's group. Genetic aging clocks will be assessed using whole blood obtained at baseline. and we will analyze 5 epigenetic aging clocks. Specifically, regulation of many DNA methylation probes is positively correlated with chronological age, a finding that has led to the creation of first-generation epigenetic algorithms, which use methylation levels to predict chronological age. Second generation of epigenetic clocks built on physiologic measures of systemic function aim to predict lifespan and health span. More recently, a new generation epigenetic clock predicting the rate of biological aging, the Duden pace, was introduced using longitudinal data on organ system integrity across 19 years. The Duden pace aims to quantify the pace of biological aging through a single time point measure of DNA methylation. Epigenetic age acceleration will be calculated by regressing epigenetic age on chronological age in months at the time of data collection, and the residuals that result will be used as a measure of epigenetic age acceleration. A positive epigenetic age acceleration indicates higher than expected epigenetic age and thus accelerated aging, whereas a negative epigenetic age acceleration indicates lower than expected epigenetic age and therefore slower aging. Descriptive statistics will be run, and there will be tests for normality in the continuous variables. Any non-normal distributions will be transformed. For aims 1 through 3, a matrix of partial correlations will be created between 4 domains of physical activity, i.e. occupation, transportation, household, and leisure time, 4 time periods, specifically school age, adolescence, young and middle adult, and 3 dependent variables, specifically amyloid burden, cognition, and biological aging, all controlling for APOE4 genotype, education, depression, and a score indicating the predicted composite risk of having a major cardiovascular event in the next 10 years. To address AIM 4, statistically significant findings will be followed up with multivariable linear regression and mediation modeling. For multivariable linear regression with a sample size of 98 and an alpha of 0.05, power is 0.83 to detect a median effect size with 4 physical activity and 4 control variables. These are my references. Thank you for your attention.
Any questions for Dr. Torres? I think we're time for a break anyway. So, correct, Stephanie?
Yes, yeah, we have a 5-minute break ahead of us, so we can stretch our legs a little bit, and I'll see everybody back here at 1:40.
At 1:40. Okay. All right. Excellent. Thank you.
Yes.
All right. Excellent.
All right. Can everyone see this?
Yes.
Yes. Great. So just to introduce myself, I'm Connie Wu. So I'm I guess still relatively new assistant professor at the University of Michigan. And so just as a brief background, I don't actually have an intro background slide, so I'll just talk. So I started here in this past January. So I've been here about 8 months now. Or 7 months now. And so I'm in the Life Sciences Institute here and also in the Biomedical Engineering and Pharmaceutical Sciences departments here. And so my background, I was formally trained as a chemical engineer during my PhD, where I was working on RNA therapeutics. And then I transitioned to more of the diagnostics field for my postdoc. So learning skills in bioanalytical chemistry, clinical diagnostics, where we were developing and applying ultrasensitive protein detection tools, looking at different biomarkers in cancer. And so for this MIDRC REC project, we're interested in applying these ultrasensitive single molecule protein detection technologies towards detection of circulating forms of tau for Alzheimer's disease diagnostics. And so my background generally has been more in technology development, also in cancer. So I'm pretty new to the dementia field. So I'm excited to use this opportunity to get more into Alzheimer's disease and learn more. And so just to introduce my lab a little. So today I'll talk more on the diagnostics side, but my lab is about 50% more on the RNA therapeutics side, and then the other half is on diagnostics, which I'll focus on today. And so basically on the diagnostics side, how I met, so my group is focused on both clinical applications of those diagnostic tools that I was talking about, and then also interested in tool development, so further improving these technologies. And so on the clinical application side, so we're interested, as I was saying, to measure circulating pathological forms of tau, as well as potentially other protein biomarkers in other diseases, as well as extracellular vesicles. So it's a very versatile tool that can be applied across many disease applications. And then on the technology side, we're also interested in really further expanding this kind of ultrasensitive technology towards improved multiplexing capabilities at such very low abundance biomarker levels. Also applications in expanding, applying some of these ultrasensitive tools to help screen for nanoparticle therapeutics as well. And so for this project, we're interested in applying these technologies using different sets of antibody pairs, affinity reagents to look at different forms of tau in plasma. And so here I'm showing you different types of phosphorylated tau assays that have been developed by various biotech companies over the years. And so for Alzheimer's disease diagnostics, it's especially promising to try to use plasma as a biofluid because it's amenable to repeated sampling. It's minimally invasive compared to a really invasive lumbar puncture to get CSF. But the problem is that these pathological forms of tau exist at extremely low levels in blood. And so you would really need very, very sensitive tools to get at such low levels. And so a lot of work is being done in the field to develop plasma-based tools to develop a blood test for Alzheimer's disease. So it's definitely a lot of work going in here, especially focused on different phosphorylated sites as shown here. But there still really remains an issue of first technology-wise issues in sensitivity. So really trying to get at extremely low levels, especially potentially during early stages of disease, where in blood it's already at very low levels. And then also really trying to get an idea of different circulating pathological forms or truncations of tau that might be circulating in blood that might reflect disease pathophysiology. And so a lot of this work this idea started when working with Nick Kanan, who's also my mentor for the MIDRC REC. So his lab has developed a whole panel of different antibodies for different epitopes and forms of tau, and also looking into applying these antibody pairs on an existing digital ELISA platform called single molecule arrays, or SIMOA. But his lab has found that we really need even higher sensitivity than even the current gold standard protein detection methods. And so that's kind of where we come in, where we've developed really, really ultra-sensitive tools to see whether we can really better detect these pathological forms of tau with high accuracy. And then more recently, I came across also another member of the MIDRC, so Pete Tessier, here at Michigan. So his lab also has developed several nanobodies and also antibodies specifically for looking at differentiating between aggregated forms of tau. And so I think it would be interesting also to integrate some of those into the panel as well. And so for the rest of this talk, I'll talk more about the technology that we'll use in this project. So that's kind of where I come in from the kind of chemical or biomedical engineering side. And so we're interested in proteins in general. because proteins are essentially kind of where the biological action is at. And you can think about all the proteins in our blood, all the proteome essentially as an iceberg, where most of it actually remains submerged and difficult to detect. So if you just think about plasma alone, 99% of the mass in the plasma of proteins is made of just only 22 different proteins. And so in that remaining less than 1%, there's actually, in some estimates, over 10,000 different proteins. And so you really need tools to be able to detect and differentiate between all those different proteins behind that mask of 99% of the plasma. That's many abundant proteins such as albumin, et cetera. And so in other biofluids such as saliva or urine, a lot of proteins of interest exist at even lower levels than in plasma. So you really need very ultrasensitive tools. So normally to measure proteins, the current kind of standard that most people use is based on enzyme-linked immunosorbent assay or ELISA. And so these are antibody-based methods that bind to specifically to your target analyte, usually done in a plate-based format. And so basically these are where you have your antibodies coated on a bottom of a plate. Then often you can use two different antibodies in kind of a sandwich format for more specificity. And so these are really nice for a lot of different proteins, but their limited detection is only as far as about picomolar or 10 to the minus 12th molar for the most part. So you can only get to a certain fraction of proteins in a proteome, but a lot of proteins still exist below this limit of detection. So we really need to be able to have more sensitive tools to uncover more of the proteome iceberg, if you will. And so where we come in is using these digital approaches. So a lot of the ELISA techniques that I was talking about on the previous slide rely on these typical analog or bulk measurement schemes. And so just to illustrate here, the difference between analog and digital is so on top we have our typical analog scheme, and let's say our readout is an optical readout such as fluorescence. And so generally as your target concentration increases, your signal intensity also proportionally increases. But when your concentration becomes too low and the number of target molecules in your sample drops too low, the overall concentration basically in that whole volume becomes too low to be able to be detectable by conventional instrumentation. And so, however, in digital approaches, now instead of trying to integrate across the signal across a whole volume, now we're compartmentalizing the volume into many, many different ultra small volume containers, such as microwells or droplets in this case. And in this way, we isolate single target molecules into these ultra small volume containers. And then now the signal that's generated from an individual molecule is at a high local concentration within each of these ultra small volumes, at a concentration that's locally high enough to be detectable by conventional instrumentation. And so in this way, now you're simply counting the number of containers or compartments that have a molecule or not. And now your signal is essentially based on the presence or absence of signal rather than having to integrate the signal intensity across an entire large volume. And so this type of digitization, this resolution that digital signals afford us really allow us to achieve much higher sensitivity in our measurements. And so you can apply these kinds of digital approaches to protein detection. So again, here just to show you kind of in the context of ELISA. So on the left is your typical analog ELISAs where you have your antibodies coated across a 2D surface on the bottom of a well and a plate. And so you rely on the diffusion of your target molecule shown in red here to the bottom of the plate for capture. But when you're at really low concentrations, such as femtomolar protein concentrations, which is 10 to the minus 15th molar, it really takes, you're relying solely on the diffusion of the target molecules to a 2D surface. And it takes on the order of months actually for the system to come to equilibrium and for to get efficient target capture. And so it's really kind of a kinetic or transport kind of barrier. But in digital ELISA, we do two things to really overcome these kinetic limitations at these very low concentrations. And so the first thing is that we use, instead of a 2D coded plate, now we use antibody coated beads that can be mixed with the solution. And so in this way, you're overcoming that diffusion barrier by essentially bringing the surface to the molecules rather than making the molecules have to diffuse to a 2D surface. And so that's the first kind of aspect of digital ELISA. So you're really overcoming that kinetic limitation by using lots of antibody-coated beads. And the second key part is that we use a high excess number of antibody-coated beads over the number of target molecules. So essentially now we have essentially a distribution, a Poisson distribution, where the number of target molecules on each bead is either 1 or 0 molecules. And so again, how we ensure that is by using a high excess number of beads. And so that's where the digitization comes in. And so you can use this. So essentially, this is a digital form of ELISA. And so what was developed over 10 years ago now is now has been commercialized and is now kind of like the current state of the art for ultrasensitive protein detection. So this form of digital ELISA is called single molecule arrays, or SIMOA. And so essentially, so you have your antibody coated magnetic beads. There are a high excess number, so you capture single target molecules onto the beads. And so most beads will carry either 1 or 0 molecules. Then you form your immunocomplex sandwich as you would in a regular ELISA with a second antibody for specificity. And this antibody is biotinylated. Then that allows you to label each single immunocomplex with and streptavidin conjugated to the enzyme. And so now you load single beads into a microwell array. And the key part here to get the digitization is that also, besides having single molecules on beads, your microwells, each microwell is can physically fit almost only 1 bead. So now essentially you're isolating single molecules into single microwells. And we also load this fluorogenic enzyme substrate along with the beads into the wells. And so once we seal the wells with oil, essentially every well that contains a single enzyme molecule will generate fluorescent signal because of the turnover of the fluorogenic enzyme substrate. And so essentially you can now count the number of molecules by counting the number of fluorescent on and off wells. And so this digitization of signal and resolution allows us to achieve over 1,000-fold, so over 3 orders of magnitude higher sensitivity than conventional ELISA for a given antibody pair. And so SIMOA can reach down to femtomolar, which is 10 to the minus 15th molar limits of detection. So this really allows us to access a previously inaccessible window of proteins. But SIMOA still has its challenges. And so one is that there's still, so now it goes to femtomolar, but in many cases, such as, tau and plasma, sometimes, especially in early stages of disease, it could exist at even lower levels than femtomolar. So SIMOA still really can't detect a lot of these pertinent protein markers in a lot of neurodegenerative diseases. So you really need even higher sensitivity. There's also limitations in multiplexing because you're limited by both cross-reactivity and also the total number of microwells in your array. And also right now, SIMOA, the current instrumentation's pretty specialized and very large. And so it's not readily accessible by, let's say, any kind of clinical or research lab. You have to really buy this very specialized instrumentation. And so during my postdoc, I was trying to set about looking for ways to improve this technology, these kinds of digital forms of ELISA. And so the key part here, what we came up with was to change the signal amplification method, where now instead of having an enzyme substrate that's freely diffusible, and so because of that, we had to compartmentalize the beads into microwells. Now we use a localized form of signal amplification of these captured single target molecules. And so again, here we capture single target molecules, but now instead of labeling with an enzyme, now we're labeling with a DNA strand and a circular DNA template. Then this allows us to carry out a form of isothermal nucleic acid amplification. So in this case, called rolling circle amplification. So we have a DNA polymerase that can essentially amplify this circular DNA template repeatedly. So travel around it in circles to create basically many repeats linked together of the same template sequence. And so then this allows us to hybridize complementary DNA sequences with fluorescent dyes attached to it. And so in this way, you generate a high localized density of fluorescent signal that's attached to every single target molecule you capture. And so this localized signal then allows us to have much simpler readouts. We don't have to put the beads into microwells or droplets anymore. And so now what we can do is essentially just read out, have a solution-based readout. So read out the beads by a conventional flow cytometry, which is often used for single cell analysis, but you can also use it for single micron sized bead analysis. And so essentially now in this way, we can read the fluorescence intensity of single beads in very high throughput manner. And so here I'm showing representative kind of plots where the x-axis is the fluorescence intensity. And so you can see that you can easily distinguish between off and on beads. And the beauty of digital ELISA is that essentially you don't really care about like the exact intensity of these beads. You only need to draw a threshold. It's essentially binary whether there is a molecule or not. And so we call this molecular on-bead signal amplification for individual counting or mosaic. And so this allows us to achieve high sampling efficiencies as well. Now we don't need to load the beads into a microwell array, which can have low efficiencies. Now we simply flow the entire solution through flow cytometry. So high sampling efficiencies also allow us to count more molecules, and so also have, which allows us then to have higher precision. Then that can increase our sensitivity. And so compared to the conventional kind of like state-of-the-art digital ELISA or Simoa, with Mosaic, we're able to achieve up to an order of magnitude higher sensitivity compared to Simoa. And we tested this across a range of representative cytokine targets. And so in some cases, we were able to even reach down to single-digit attomolar, so 10 to minus 18th molar protein concentrations. And just to kind of give you a sense, that's in like a typical 100 microliter sample volume, 6 attomolar. So this is only a few hundred target molecules. So we're really able to get the sensitivity to that kind of low level, which is obviously still dependent on the antibody, but for a given antibody pair, now we're able to push the limit of detection even lower. And so now we're able to access even a larger window of proteins. And so we've applied this Mosaic technology to different markers, biomarker candidates in cancer in my previous work. And so now I'm interested to see whether we can apply this to different proteins, biomarkers in neurodegenerative diseases. And one thing I forgot to mention here is that also Mosaic, because it uses a solution-based signal readout, we're also able to improve the multiplexing capabilities, where previously in microwell array-based methods, we were limited by the total number of compartments where we use different fluorescent dye-encoded beads for every target. But now essentially you can flow an unlimited number of bead types in theory in Mosaic, and this can really expand the multiplexing capabilities, which I won't go into too much detail here in the interest of time. And so just to recap, it's more, so Mosaic both gets more high throughput, sensitivity, multiplexing, and also accessibility using conventional instrumentation. And so now we're interested in screening different antibodies and affinity reagents for different pathological forms of tau using Mosaic, potentially then looking also into multiplexing and then testing in a pilot cohort of plasma. And so what we're starting with here, just as an initial set of assays from, we're using some antibodies from the Canaan lab, and they've developed Simoa assays using these antibodies, looking at different epitopes for different antibody pairs. So essentially recognizing normal tau, different kinds of truncated C-terminal tau forms, et cetera. And really trying to, first we'll develop assays and then compare to Simoa and look at their performance in plasma. Then we'll expand into multiplexed Mosaic panels for simultaneous detection of different tau forms in plasma, and then finally evaluate the diagnostic performance in a pilot cohort of patients. And so hopefully this will set the stage then for larger cohorts if this works well in plasma, and then also looking at other forms of many different forms of tau using different antibody pairs using large multiplex panels. So with that, thank you very much. Thanks to Nick for helping me with the application, and then also current lab members for building up the lab. So thank you.
Very good. Any questions for Dr. Wu? Okay, let's go on to our last talk then. I think, is it Wei Chen or do you prefer Arthur?
Yeah, I can go with Arthur.
Okay, well, Dr. Zhu, please go ahead then.
Sure, let me share my screen. Guess you can see my desktop here. Give me a second. Can you see my slides?
Not yet. There you go.
Do I have to swap or like it's the regular view here?
It looks fine.
Okay, good, nice. Today is my great honor actually to be here as one of the mentee in the Michigan Alzheimer's Disease Research Center REC program. And I'm very glad to share some of my current work and also introduce myself as well as the proposal I put in the REC program. Before I introduce my research, actually, I really want to introduce my regular research interest. So basically, my primary research interest focused on the discovery and analysis of the genomic and genetic variations in the human population, as well as its impact on the population diversity and the various of genetic disorders. And by combining the bioinformatics pipeline with the data from variable genomic technologies, basically we can explore the origins and the distributions and also the mutation process of these genomic and genetic variations in both the healthy samples as well as the patients with the disease. I involved into a lot of like international collaborative consortium, and one of the consortium named BSMN, which is the Brain Somatic Mosaicism Network, is my first experience that I started some study related to the brain. And basically, we explore the brain somatic mosaicism of all of those genetic variations in different brains of regions. And more recently, I joined the SMART consortium, which is the somatic mosaicism across human tissue. It's not only focused on the brain, but also on the 30 tissues across the human body. And with all of this consortium experience and during my time of postdoc training, I'm able to develop those both experimental technologies and as well as the bioinformatics pipelines. And I'm trying to bridge the gap between the cryptic genetic variations and the human development as whereas the neurodegenerative diseases. I found like one disease is particularly interesting is the also aging related, it's the Alzheimer's disease. Therefore, currently I'm trying to leverage my expertise in the Alzheimer's disease field. And so here's some like my recent related funding work, basically related to the Alzheimer's disease. We have a R21 that's supported by NIA that collaborate with Alan Boy and Dr. Ramios and Peter Todd in the Department of Neurology. And basically we explore the repeat element in the human genome and relate in the Alzheimer's disease samples. So there are more recent, there's a publication that we put in the bioarchive that we want to explore the distribution and the accumulation of the somatic nuclear mitochondrial DNA insertions in human brain. That makes me think that probably that would be the starting point that I can start to try to fill the gap between the nuclear mitochondrial DNA insertions and Asian-related neurodegenerative diseases, especially the Alzheimer's disease.
Just some background before I introduce some results. I believe everybody is familiar with the mitochondria and the mitochondrial DNA. Basically, mitochondria is the energy factory of the cell in our body, and this function of the mitochondria will raise the oxidative stress level, and eventually will lead to some link to some aging process. Subsequently, will potentially lead to the age-related diseases. So the human mitochondrial DNA include about like 16 kilobase pair nucleotides, as well as encoding 13 proteins here. And what is NuMite? As I show in the figure on the left here, basically NuMite represents the nuclear mitochondrial DNA insertion, which is basically here we have the mitochondrial DNA, and it leaks from the mitochondria and inserted into the nuclear genome of the human cells. And this NuMite have already been observed in many organisms, and it also reported related to the cellular evolution, function, and the many cancer diseases. More interestingly, actually, it was reported by several publications, basically saying this NuMite are correlated with the chronological aging in the yeast and the rats. Therefore, just step back a little bit, about since we focus on the brain and Alzheimer's disease. So basically, actually, while I'm doing the somatic mosaicism network, we are focusing on the somatic mosaicisms in the brain, and the somatic mutation, we observed, would already reported to raise accumulated during the human development and the cell, also the brain neurons, and also more specifically to the Alzheimer's disease. Actually, a lot of studies already reported the specific single nucleotide variation, or we call CNV, in the hippocampus, as well as the copy number variations in neurons that contributed to the progression of the certain sporadic Alzheimer's disease cases. And especially the ones that the copy numbers related to genes such as APP and ApoE that already be studied very well. However, the genetic factors contributing to more than half of this remaining Alzheimer's disease cases is still unclear. So I ask a question: are the NuMites a possibility mitochondrial DNA nuclear DNA signal accesses in the human aging and then potentially aging-related neuron degenerative diseases, especially the Alzheimer's disease? So I have our hypothesis basically here is we want to we hypothesis that the somatic NuMites were accumulated with the age in the brain or certain brain regions, and eventually we could have some potential role and functional impact on the Alzheimer's disease and Alzheimer's disease related dementia. The object that we studied here is the RoseMap project, RoseMap data. The RoseMap content actually include two cohorts. The one is called Rush Memory and Aging Project, which is the MAP, and the other one is the Religions Order Study, which is the ROSE. And overall, the RoseMap have 1,187 samples from different individuals that we included, and also for all of those 1,200-ish samples, we have the different tissues, either from the post mitotic brain tissues from different brain regions, or the circulating immune cells, either from whole blood or other immune cells. And also we identifying all the patients' age at death, as well as the dementia status based on basically it was coded on the non-cognitive, as I showed NCI here, or mild cognitive or Alzheimer's disease dementia, from the final clinical diagnosis of the dementia. And the Alzheimer's disease classification was defined based on the National Institute of Aging Reagan criteria. Since we have the roadmap data cohort here, so the next step, basically, I want to identify the pneumat or the somatic pneumat among those samples. This is the pipeline basically I developed to identify the tissue-specific pneumats in the roadmap cohort. To tell a long story short, basically, we have all the short-read whole genome sequencing data, and we use in the package and compare to the across the samples, as well as to move the background noise. Eventually, we will get the tissue-specific pneumats in from each individual samples, and then that would be potentially the somatic pneumats. So here are some results I want to share here. First one is that we observed a enrichment of somatic pneumats insertions in the DLPFC samples. So as you can see from this figure here, basically the x-axis is the different brain regions as well as the whole blood control samples, and the y-axis basically is the pneumat count per individual. And all the dots are the pneumat counts in each individual. As you can see that actually we observed a way more somatic pneumats insertions in the DLPFC samples compared to either the cerebellum or the PCC or the circular immune cells. Then I further asked a question: does the pneumatic distribution correlated with age or cognitive status? So based on I mentioned before, so we have all the information of the patient that the age at death as well as the cognitive status. We break down all the samples for the DLPFC sample into 3 different cognitive group: NCI, MCI, and Alzheimer's disease. And what we observed here, basically the DLPFC specific pneumats are negatively associated with the age at death in person without cognitive impairment, which meaning that basically the DLPFC tissues of individuals with no cognitive impairment who died younger at younger age carry about 2 more pneumats per decades of life lost than those who lived long. We also observed a similar a little bit lower negative association in the MCI group of the DLPFC samples. But this is not very significant, and we do not observe any significance there like in the Alzheimer's disease sample. And then I asked a question, so what about the other tissues? And so I investigate the brain regions that with the enough sample size. And luckily we have the enough sample size for the cerebellum samples. For the cerebellum, actually we observed the same trend for for all 3 groups. Interestingly, we observed a weak positive significant associated between the age between the pneumat accumulation or pneumat count and the age at death in the Alzheimer's group in the cerebellum group, in the cerebellum samples. However, I would say like this correlation is not very significant given the p-value here. So by combining all of this observation together, actually this emerging evidence has shown a connection between the Alzheimer's disease aging and the pneumat activities in the post-mortem brain samples. And this requires a further investigation for a wider range of data samples and more extensive details. Therefore, I propose the proposal here basically for the RAC program. I have 3 specific aims. The first aim here is to explore the Alzheimer's disease-related somatic pneumatics using genomic and the phenotypic data from large cohort. Here, the large cohort, I mean the NIA genetics of Alzheimer's disease data storage site, or called NIAGAS, since we have already granted the access to this big, huge data set. And the second aim here is to discover the expression level of the pneumatics and using the QTL, or the quantitative trait loci analysis associated with pneumat and across the different regions of brains in the RoseMap data. Since we have the RNA sequencing data of matched sample in the RoseMap cohort at part of the samples, the sample number is 487. So we can use that to do the RNA sequence analysis and also the QTL analysis. So these are some introduction for the NIAGAS database that we're gonna use for the M1. And as you can see, the table here, it include a lot of cohorts, and including the RoseMap cohorts. And you can see like overall we have like 56,000 sample that either Alzheimer's disease or Alzheimer's disease related. And one of our previous challenge here is basically to go through all the administrative process and to have the access to the database. It costs us like 1.5 years to do the application, but eventually now we have the access. And surprisingly, we had the overall sample for all of those samples. It has like more than 1,000 terabyte size for all the WGS whole genome sequencing data. So the current challenge here for me right now is to implement, like how to implement our NeuMap pipeline on this data set in and obtain the overall landscape of pneumats of all of the across all of these samples in a practical way. Because by using the traditional method, actually, if we download it and archive in the Great Lakes of the UMich or some computing cluster, it will cost a lot of time and a lot of money. So luckily, our group have figured out a way to basically utilize the Amazon Web Service to do the analysis. It's basically on cloud computing infrastructure. Here is like a schematic how we can implement this. Basically, this dedicated pipeline allows us to automatically schedule and execute the NeuMap pipeline on the AWS cloud, and to call the NeuMats from the whole genome sequencing data, the metadata of like those terabyte level data in the NIAGAS. And yeah, pretty much that's pretty much it. The expected outcome here, basically, I expected to establish a set of the somatic pneumats in the NIAGAS cohort. Actually, we have already run a pilot run, and we get some preliminary data, and the pipeline was proved to very efficient and save us money. And that if we could find that set of the somatic pneumats that could correlate with the age or the cognitive status. And then the second one, I would expect to have a list of pneumats that with the correlation on the gene expression profile with the in the different regions of Alzheimer's disease brain and other clinical information that would potentially give us some clue that the NeuMap plays some functional impact, a role or the impact on the Alzheimer's disease or Alzheimer's disease related dementia. And for the future direction, actually, I would love to learn from the expert in the Alzheimer's disease field, since I'm really new to the field. And I really want to do some more communication with the group, and also maybe better interpretation of all the clinical information. Since right now I really don't know how to handle all of those meta data from the NIRGAS in terms of the clinical information. And the second one is like more ideal was if we can have some samples that with the multiple layers of omics data, matched omics data and the clinical information, or even have some samples that we can do the, I can do the sequencing in-house in our lab, so that we could have a better systematic analysis locally to investigate the impact of the pneumat or the other genetic variants in the impact on the Alzheimer's disease. That would be perfect. With that, I would thank you for your listening, and I would take any questions and comments.
Any questions for Arthur? Okay. With that, why don't we go on to overview? I think Stephanie, should I share my screen, or are you going to show the slides? There we go. So we're just going to go through a quick overview of the ADRC and then just highlight some of the potential resources the ADRC has. So let's go to our first slide. So next slide, Stephanie. We're frozen. Oh, here we go. Yeah, so I think the ADRC consists of a number of cores and an overall administrative core. And this is just the table of organization. So you can see there's a clinical core, neuropathology core, data management statistical core, biomarker core, neuroimaging core, outreach recruitment engagement core. All of those, to some extent, we interface with all of them as part of the REC, and they're all overseen by the administrative core, which is run by Hank Paulson, Bruno Giordani, with the advice of a variety of advisors, executive committee and external internal advisory board, and then an advisory council, right, because of the strong prominence of the Center of Recruitment Activities in the Detroit area. Next slide, we'll just go over some of the individual cores now. The administrative core, this is the core that's responsible for the overall administration of the center. Hank is the director, Bruno is the associate director. Ari Baumick, who's on this call, is one of the research administrators. Nancy Larissy handles most of the business matters. She's the person you'd be dealing with most commonly in terms of financial issues and support. There's a number of staff members, including regulatory specialists and DAC coordinator. And then Peter Lichtenberg is also involved in the administration core because we have a strong presence in Detroit. And the goal of this core is really to make sure that all the other cores run smoothly and interact with each other, and this general center program gets carried forward. Just to mention, an important component of our center is specifically trying to bring forth junior investigators, and talk about that a little bit more in a minute. Next slide, please. So the clinical core, this is really probably the most largest core in any Alzheimer's center. This is the core that's devoted to the recruitment and characterization of research subjects. And the clinical core follows a substantial number of individuals who are in a very longitudinally followed cohort for whom there's a very rich data set with increasing a lot of imaging and biomarker data. But in addition to that, there's also a somewhat separate research roster, which is somewhat larger, which is also accessible to potentially access research subjects. And this core is led by Ben Hamstead and Judy Heidebrink, and Steve Campbell is the core manager. And there's a number of personnel who are involved in subject characterization and recruitment. And again, this core exists in large part to characterize and follow individual research subjects, and is the place you would go to try and get access to research subjects. Next slide, please. The data core, which is led by Kelly Bukulski, and John Reeder is the core manager. This core runs our database for all the accumulated data that's come forward. This core also offers a significant amount of expertise in terms of statistical expertise for consultation, advice on the design execution of experiments, and staffed by a number of very, very competent statisticians and data analysts. So again, this is, I think, all of our cores are strong. I think this one is particularly strong in terms of data management and analytical ability. Next slide, please. And the neuropathology core, this is a core that's devoted to characterization of pathologic samples from some of our longitudinally followed subjects, as well as maintaining a brain bank for research purposes. Andy Lieberman, who's the head of neuropathology here, is the director. Matt Perkins is the brain bank coordinator. Again, there's a number of other individuals who are involved both here at Grand Rapids as well. And this is an excellent resource for accessing tissue, not just on Alzheimer's disease, but on a variety of neurodegenerative disorders. And in addition, all the brains that are received through the brain bank get characterized in a very systematic fashion. So there's a very rich data set, all of which goes to the National Alzheimer's Coordinating Center in Washington State, the NAC. Next slide, please. And this just shows you a recent inventory of what's in the brain bank. You can see there's a wide variety of different types of specimens, including control specimens, ALS, some Parkinson's disease, Huntington's disease, and a large number of Alzheimer's disease and related disorders. Next slide, please. And then a new core in this cycle is the neuroimaging core, led by Doug Knoll and Ben Hampstead. And this core is again devoted to acquiring and analyzing imaging data, including both MRI and PET data. Again, the personnel on this core really have outstanding expertise in terms of the analysis of the imaging data, including people with, I would say, international reputations in this area. So again, this is another very useful resource for research applications. Next slide. And then another new core, the biomarker core, led by Nick Kanan and Dave Morgan in Grand Rapids, again with some additional individuals. This core is devoted to really looking at peripheral blood biomarkers for discovery and validation. You heard Dr. Wu briefly touched on this briefly in terms of some of the reagents that are available from Nick Kanan's laboratory. But there are other potential reagents as well as quite a bit of technical expertise in terms of the Simoa assays and mass spectrometry. And I think next slide. And finally, the Outreach Recruitment Engagement Core. This is a core led by Scott Roberts in the School of Public Health. Core manager is Renee Gadwa, who I believe is still on. In any case, this core is really devoted to outreach, partly for education in general, but also for recruiting of subjects. And again, this is a very strong core with a number of individuals at all of our sites involved in this core. Let me just mention that in every single one of these, I think almost all of these cores, there's usually a younger faculty member, several of whom have been prior REC mentees. So we have sort of a pipeline going from the REC into more intense involvement in the Alzheimer's Center, and then with the ultimate goal that some of our trainees will actually graduate to become not only independent investigators, but potentially core directors as well. Next slide. And this just is some of the other initiatives that are associated, supported by the Alzheimer's Center. It's a Lewy Body initiative led by Renee. This is supports some support groups around the state. And we have a Lewy Body Dementia Research Center of Excellence. And then a wellness initiative, particularly for caregivers, led by Laura Rice-Osugar, which again is a really excellent outreach activity for providing support education about dementia and for wellness for caregivers in particular. And next slide. And finally, the REC. So there's one of us at all of the three institutions, myself at Michigan, Peter at Wayne, Irving Vega at Michigan State. Irving unfortunately couldn't be here because of a family emergency. And most important, Stephanie Nava, who is the coordinator and is definitely the most important person in the REC. It's the person most of you will be dealing with. And again, our goal is to try and help you, grow your careers in dementia research, partly through some financial support that we're providing, partly through some enhanced mentoring, and partly through some of the resources that are available through the ADRC cores. Next slide. And Peter's going to talk about available resources.
Thanks, Roger. you know, it was great to hear these presentations. Several people who are just coming into Alzheimer's disease research were thrilled that you've decided to join that because it's so needed. And several people already are making good use of our resources because of their mentors. So let me just go through a little bit some of the things that our center can provide to you. We heard a lot about some big data that people are using in their studies. And so, you know, that data core can be very helpful to you for sure. I know some people are using the MADRC longitudinal data set, and the clinical core is really going to be a big connector so that you understand everything that's in there and getting a hold of it through John Reader and so forth. And we heard about several neuroimaging projects, and of course, the neuroimaging core. And Nick is here. Nick has been with us the whole time. Thank you, Nick. And we're thrilled with this core, and it's so great that we have so many projects that are really going to interact with this core and our biomarker core. And you know, that's also a very strong core. Roger mentioned the neuropathology core. Didn't hear any particular projects for the rec studies that are going to be using that core, but there's some great opportunities there. I really encourage you to reach out to the core leaders where it's relevant to your field of study. I also really want to encourage you to, and maybe this is on the next slide, and we'll come back to it, to really look through the very carefully selected lectures that we have on Alzheimer's disease. It's a very good introduction. And several people here are coming in through cancer or diabetes, other kinds of areas. You're going to find it very useful. We have an annual co-sponsored Pepper Center Career Development Retreat. That's an opportunity. We always have an annual Beyond Amyloid Research Symposium. Next one is going to be in Grand Rapids or East Lansing, May, June of 2024. We'll have to get that date settled down pretty soon. And the opportunity for developmental projects, and Arthur has one. And this is $50,000 to carry out a more robust study. And so look for those application periods. You know, even if not this year, maybe next year, when you really had a chance to get some of your data down and are ready for. We have some travel awards to get to conferences, and that's kind of announced as we have those conferences come up and the monies. And a really interesting series, clinical pathological correlation conferences, where we hear about individual cases, including their brains at autopsy, but we hear the whole clinical picture, and we get a real sense of kind of how tricky these diagnoses can be. So it's an outstanding series, and Stephanie will alert you to those as they come up. They're usually a Friday at noon, and well worth attending. Next slide, please. Yeah, this is the online dementia curriculum. This is really, and this was a great innovation by Roger. He really encouraged us to do this for our first 5-year cycle. And we are redoing some now in our second cycle, which is great. But it's really useful. These sessions are about 20 minutes, 15, 20, 25 minutes at the most. So they're really in bite sizes that you can really make use of. And I really encourage you, especially folks who really are coming with little background Alzheimer's disease, to get a sense of the broader field and what people talk about, because this is being approached from an interdisciplinary perspective, and this curriculum is certainly that. Next slide, please. Those are the resources. What's the total expected time? That's a great question. Stephanie, what do you think? If was it about 25?
Yeah, it's about 25. Yeah, it's about 25, and they're, as Peter said, they're 20 minutes. I don't think I think the longest one is probably half an hour.
Yeah,
and that most of them are closer to 20 minutes. So, and
I'm
you
gonna,
can
I'm
spread
gonna
it
say
out.
10 to 12 hours total.
Yeah, something
Yeah.
like, yeah.
So that, and the bite sizes really helps you do that because as Roger said, none of them go over a half hour. And so that could be great.
No, the answer to Dr. Torres' question about continuing education credits, no, not at this point. But you can get continuing education credits for the CPCs.
Yes, great point, Roger. Thank you. So the clinical pathological conferences do offer CEs. Okay, let's take a look at some of the expectations then. Stephanie, next slide, please. Now, hopefully you've all selected a mentoring committee. So you have a primary mentor, but we want you to have a committee with 2 other people as well. Your frequent meetings are going to be with your primary mentor. And we really want you to make sure that you do this. And if you're having any trouble with it, let us know. So, for example, Norman, you would contact Irving because he's at MSU, probably knows. Yeah. And Elisa, you would contact me. I'm here at Wayne State. Rest of you contact Roger. If you're having some problems, we can assist with making sure. But from our feedback, from former rec trainees, that's the key. The primary mentor relationship and meetings, and getting the mentoring committee to meet every 6 months. You might want to scope out an individual development plan for yourself, what your research goals are, any training that you need to do, etc. You will have regular progress reports to the rec. And as Roger says, that's Stephanie. She will be communicating about that. We of course will look at those carefully, but Stephanie is the one who will be alerting you to that. Every year we'd like you to meet with your on-site co-leads. So as we just mentioned, people at the same university. The annual presentations at the MADRC Symposia. So we'll expect folks to present. Even if it's a work in progress, we still want, that's fine. But we really want our rec mentees to present. Stephanie, there's a great question. How often are the progress reports? I think they're every 6 months, aren't they?
Yep, they're every 6 months.
Yeah.
And then the final time will be like a different closeout. So there'll be 3 that are exactly the same format, and then their final one will be a little bit different.
Yeah. So not a heavy burden.
No.
Not at all. But it helps you and it helps us.
Yeah.
So that
try to time them after you have one of your committee meetings, essentially.
Yeah.
Yep. And I'll be in touch with you to make sure you get all those things scheduled up. And yeah, they're a really great way for us to keep up with all of you guys and what you're doing, what you're publishing, what grants you've applied for, any sort of troubles you might be having that can be brought to the rec lead's attention. So they are very helpful.
And again, we encourage you to use the MADRC resources. That's what the MADRC is here for, to help generate more and more research on Alzheimer's disease and related disorders. Next slide, Stephanie. So I think I covered most of this. People don't need a refresher on what to do with a primary mentor, I think, given our group. And the committee members, though, can really also be great promoting a mentee's career advancement, getting contacts that you may not have thought were important that become important, as well as kind of assist with grant applications, particularly. Next slide. So we expect to meet annually with the mentees, And we'll be taking a look at these 6-month progress report and looking at your career development research project, and also at the mentee mentor interaction. Sounded to me like a lot of you are off to a good start with your primary mentors already. And so that's often the case. And a lot of those primary mentors, if I heard it correctly, I was keeping track right, are connected to the center. And that's usually a very strong indicator of smooth sailing because people are very committed to the center and the work of the center. So it's not just about your research project. We're also concerned about your career development. And all of us are here to assist in that. And now I turn it over to Ari.
Thank you. Thank you, Peter. Again, really happy to see this incoming class of the rec mentees. Stephanie can go ahead and send the next slide. I think, there's a really heavy one out here. So as Roger mentioned earlier, Nancy and I co-administer the center. So for more of your funding questions and things like that, Nancy will be working with you directly, but I do sit in those meetings out there to make sure that your statements of work and your funding and dollars are sent to your respective institutions in a timely fashion. The rec funds will be dispersed by the end of August 2023. Those come from NIA, and so they're funds that are linked with our center out over here. Your project funding dates are from 7/1/23 to 6/30/2025. There is $25,000 in direct costs. 20,000 is actually coming from cost share funds that don't have any indirects out there. And 5,000 of that is coming from the NIA sponsored fund side. So you do get a little bit of indirect cost that is provided on that 5,000, and it is 8% per the RFA that is listed out there. So at the end of the day, you will get a total funding of $25,400 out there. For U of M rec mentees here, you will be provided 2 short codes from the neurology department finance team. One is the sponsored and one is the cost share with the same project grant numbers. The total funding, as I mentioned, is $25,400, may be spent in year 1 if needed. Any unspent funds will be carried forward to your year 2. For the Michigan State and Wayne State mentees, subcontracts can only be set up 1 year at a time. Half of the funding will be allocated in year 1, and the remaining funds will be awarded in year 2. You may overspend in year 1 in anticipation of your year 2 funds. However, the institution does not allow overspending. If it doesn't allow you, please contact us if your year 2 funding is needed sooner. Any unspent funds in year 1 will carry forward into year 2. So again, definitely reach out to Stephanie, Nancy, and we will take care of it from there. Next slide, please. Use of rec funds. I'm sure you have already read this in your applications and meetings there. You can use them for several of these things, including data analysis, effort for studies personnel, acquisition of initial data that is a departure from your primary line of research with your mentorship committee approving that and then running it through our rec leadership team out here. Any method development, equipment purchases, imaging or similar services. So very broad ability for you to use your rec funds. For senior postdoc fellows, salary support to defray work on any novel project also there. Again, other users, if justified appropriately, I would definitely recommend you reach out to Roger, Irving, and Peter to make sure that's there, and then they will loop Nancy in, and then we will sort of readjust to your statement of work as needed. Just to note, expenses which do not comply with uniform guidance rules are unallowable. Examples are hosting, postage, telephone, general supplies, administrator salary, and there is no tuition support available through these rec funds at this point of time. Next slide. For any of your posters, publications, and presentations, if you're using data from the Michigan Alzheimer's Disease Center, we ask all our rec mentees and everybody who uses the data to acknowledge the center with the grant number that's listed here and also the University of Michigan Disease Center, which is which has that Berger endowment, which is actually paying for a large portion of the REC funding here in any of your presentations, posters, and publications there. If you need any particular language, we also have it on our website. Please feel free to reach out to Stephanie and we can provide you the logos and things like that if you need to put for your posters and your presentations there. We have a whole set of approved logos there. And for publications, please apply for your PMC ID numbers, because those do connect to the grant at the time of our continuing renewal, our RPPR. And if you don't do that, you will get an email from Nancy asking you to do that out there. So you have been warned out here. So thank you for paying attention to that. And do let us know when you have a new publication. Sometimes the publishers do say they will do that, but they could take up to a year to do that there. It is a very easy process. Nancy has it written down very well. We can provide you that information as a step-by-step to actually obtain those PMC ID numbers on your own. Next slide. Other requirements to send to the administrators, if you haven't already done, your post-award specialist contact info, signed an acknowledgement statement of work form that Nancy should have sent over, a completed budget and budget justification, and copies of any of your, if you're using animal or human subject or exemption notice, if you have, please provide those there, because given it is connected to the NIA, we do have to keep track of those kind of things out there. You can send it directly to Nancy and me, and we can make sure it gets to the right place. Next slide. Okay, questions, and you have all our contact information, email, telephone, and I think that's out there. I think that's the end of my slides, right, Stephanie? Thank you.
Let me just add one thing. So all of you submitted a budget with your applications. Now, we have had situations come up where people wanted to redirect their money. Again, we're very flexible. We just want to make sure that it's justified and allowable under the federal rules. So if something happens, like you get an additional grant and you want to use the money in a somewhat different way than originally described, contact us, ask us. We'll ask you to do a short budget, redo the short budget and budget justification. But all these requests we've dealt with in the past have been very reasonable. So we do have some flexibility with these funds. Okay.
Go ahead, Andrew.
A couple of questions. Will the slides be sent around just so that we have them?
Yes, they will. We'll send the PDF of the slides around.
Perfect. And in terms of budgeting, remind me, I think we're not allowed to put our own salary on it. I'm just sort of anticipating a potential where, like, my temp grad student leaves early and I'm like, still have a little bit left. What do I do with it?
Right. You're not allowed to, faculty are not allowed to use them for salary.
Okay, thanks.
Okay, yeah. But again, if something like that happens and there's a reasonable other use of the funds, let us know. We'll just ask you to do a modest budget justification. All right.
Good.
All right. Nick, thanks for joining. Any other questions? Stephanie, anything you'd like to add?
No, I think that's it for today. Thank you very much for presenting your work. It was really nice to meet you guys all in kind of person. So.
Super. Well, that was a lot of fun talks. Thanks so much.
Thanks, everybody. Have a great afternoon.
Thank you so much.